Comparative differences in general characters of fungi. So far, the published videos all are related to general characters of fungi. So now the comparative differences between various kinds of terminologies or maybe different kinds of things that we have explained in general characters of fungi. Here I have placed some comparative differences for easy understanding as well as for preparation of exams also. I'm Anish Shankar Reddy, PhD Plant Pathology. It starts with comparative differences between Conidia and sporangia. So I have given some important things related to this. So starts with the definition. Generally, this Conidia is asexual non-motile spores that is produced by the fungi. Or when it comes to sporangia, it is asexual fruiting body that is actually present in lower fungi. For example, you can see Oomycetes and all those kinds of fungi, and which are actually formed on conidiopores exogenously. It's a very important things. So here to understand, you need to understand two things here: what is conidiopore and what is sporangiophore. That I will explain in the later slides, because in the later slide, next slide, there is a diagrammatic representation, which are produced exogenously. Exogenously in the sense outside. There is this conidia is actually produced outside. There is no packing. Uh, like inside as like of sporangia, where sporangiospores are produced inside the sporangia, which is actually endogenous. So when it comes to conidia are exogenous, which means which are produced outside. That I will explain in the later slide. Somatic hypha, that is actually produced in the endogenously. Generally, these conidia are barrel shaped, sometimes clavate shaped, sometimes flame shaped, or sometimes club shaped. Generally, this sporangia is globose, round, or generally in lemon shaped. So lemon shaped sporangia is actually we can observe in Padapthora. And when it comes to germination, so this conidia germinate directly and produces gem tube. And when it comes to sporangia, this sporangia produces sporangiospores. Small tiny spores are known as sporangiospores, so which is they also germinate directly or indirectly. And the behavior, they cannot behave like a sporangia. This conidia cannot behave like a sporangia. When it comes to sporangia, occasionally behave like a sporangia. And types, micro and macro conidia can be observed. For example, you can see fusarium. Fusarium can produce micro and macro conidia, right? And also chlamydospore. This conidia can act can behave like a both micro and macro conidia. And there is no micro and macro sporangia. Such types can be observed in sporangia. And this conidia production especially observed in Ascomycota and Basidiomycota group of fungi, whereas Oomycetes and Zygomycetes we can observe this sporangia. So this is a very clear understanding. This is a sporangia. This is a conidia. So this stalk-like structure is known as sporangiopore in sporangia. The same stalk-like structure is known as conidiopore in conidiopore in conidia. So the only difference is if it holds a globo structure, that is sporangia, the stalk-like structure is known as sporangiopore. They holds conidia. When it comes to conidiopore, that's why it's known as conidiopore, right? So here we can see this is a globo structure. This is known as sporangia, where small spores are packed inside. That is called sporangiospores. That's why it is called as endogenous. Endo means inside. When it comes to conidia, conidia are born outside, are produced outside, right? That's why it's called exogenous. Exo means outside. So here the sporangiospores can generally observed as we discussed Oomycota and Zygomycota. But when it comes to Ascomycota, higher fungi like Ascomycota and Basidiomycota generally produces. The next one, difference between Arthrospores and chlamydospores. Generally, these arthrospores are vegetative cells that have passed into the resting state. When it comes to chlamydospores, so chlamydospores are thick-walled, large resting spores that is produced by various kinds of fungi. So the next one is formation. So generally they are formed by the breaking of fungal hyphae. So you generally imagine this is the fungal hyphae that breaks into small fragments and starts to behave like a spore, that is called arthrospores. Arthrospores are sometimes known as oidea also. Here the chlamydospores are formed by the enlarging, rounding and thickening of the cell wall or terminal hyphae. Sometimes they may produce intercalary or terminal, which means, for example, this is hyphae. So here bulging in between the hyphae is called intercalary. Terminal in the sense at the end of the hyphae, if it is the chlamydospore are produced, it is terminal. If in between hyphae it is produced, it's called intercalary, in between. Okay. So thick walls, no thick walls can be observed in arthrospore, but when it comes to chlamydospore, thick walls can be observed because chlamydospore is a resting spore. To survive unfavorable environmental conditions, it required very strong thick wall. So that thick wall can be observed in chlamydospore. Pigmentation in walls, no pigmentation can be observed in arthrospore, but when it comes to chlamydospore, pigmentation can be observed. Fragmentation, yes, a fragmentation phenomenon can be observed when it comes to arthrospore. And here when it comes to chlamydospore, they are formed just before the hyphal fragmentation, before hyphae is fragmenting. So this chlamydospore is just formed before fragmentation, and also this chlamydospore is formed during unfavorable environmental conditions to survive in the soil. And whenever the onset of favorable time or favorable environmental conditions, this chlamydospore again starts to germinate. And separation from hyphae. So here separation from hyphae during the formation. So during formation, this separation can be observed. Actually, these chlamydospores are released after the hyphal death or during a very, as I told you, unfavorable time. So that time these chlamydospores are formed or released. This is diagrammatic representation. This is hyphae. This hyphae breaks up into small fragments. This fragment starts to behave like a arthrospore or arthroconidia. So when it comes to chlamydospore, we can see at the end chlamydospore are formed. As if it is forming at the end of the hyphae, it is called terminal. If it is forming in between the hyphae, it is called intercalary. So we can see terminal, intercalary. So this is intercalary. So such kind of chlamydospore we can observe, right? So this is about arthrospores and chlamydospore. Now, asexual fruiting bodies. In general characters, we have discussed asexual fruiting bodies and sexual fruiting bodies. So there are four types of asexual fruiting bodies: are Pycnidium, Sporadium, Acerula, and Cinema. So this four types of asexual fruiting bodies. General description. Generally, this Pycnidium is a hollow globose flask-shaped fruiting body. So when it comes to Sporadium, cushion-shaped. When it comes to Acerula, Acerula is a saucer-shaped. Cinema is a aggregation of conidiophores that is parallel to each other. This pycnidium is produced by the fungi like Macrophomina, Diplodia, Botrytis, Phomopsis, Philosticta, Ascobita, and Septoria. Whereas Sporadium is produced by Fusarium, Epicoccum, Strumella, Tubercularia, Voltella, Myrothecium is produced Sporadium. Acerula is produced by Culrotricum, Pastolotia, and Gliosporium. Whereas cinema is produced by Ceratocystis, Graphium, Arthrosporium, Isaria, Hypographium, and Trichoras. These are all the various kinds of example or genera that is produced from this asexual fruiting bodies. So to one, like in a comparative way, we can describe this pycnidium as a hollow flask shaped, Sporadium hemispiracular barrel shape, sucker shaped acerulus, loose aggregation of compound and branched is cinema. Generally, when it comes to the shape of the fruiting body, Pycnidium is a flask-shaped, Sporadium is a cushion-shaped, Acerula is a saucer-shaped, but Cinema is look like character branches. Examples have been given. These are all the diagrammatic representation of Pycnidium, Sporadium, Acerula, and Cinema. To have a in detail understanding, please refer our previous lectures. So where I have mentioned microscopic images also for our easy understanding. Now the difference between spores and gametes. So generally the spores are independently germinate from the germ tube. When it comes to gametes, so gametes should fuse with another gamete to form zygotes. So this kind of gametic fusion can be observed in Zygomycota fungi, Zygomycota group of fungi, right? So then reproduction, both sexual and asexual reproduction, the spores are generally produced. These gametes are produced only during the process of sexual reproduction. The production in the sense produced during asexual reproduction, these spores generally, two kinds of spores are there: sexual spores and asexual spores. In sexual life cycle, sexual spores are produced. In asexual life cycle, asexual spores are produced. When it comes to gametes, gametes are for two types: male gametes and female gametes, generally involved in the sexual reproduction. When it comes to the nature, the spores are generally haploid, which are actually born by the saprophyte. And when it comes to gametes, gametes are born by the gametophyte, which are always in haploid in nature. Both are generally haploid in nature. And spores, for example, you can take sexual spores like Zoospore and zygospore, which are diploid spores. Unfavorable conditions, how the spores will do, they're capable of surviving unfavorable conditions. When it comes to gametes, pollen can survive during unfavorable environmental conditions, so other kinds of gametes they cannot survive. So morphological types is homosporous and heterosporous. When it comes to gametes are isogamous and anisogamous, and the cell wall is mostly surrounded by very thick cell wall for surviving harsh environmental conditions. When it comes to gametes are naked, generally covered with a very thin cell wall. Thin cell wall, they cannot survive during unfavorable time. Animals do not produce any kind of spores, whereas these gametes are actually produced by animals. Coming to the difference between zoospores and aplanospores. So this zoospores, in the previous lecture itself, we have discussed very in detail about this zoospores. Generally, these zoospores are motile asexual spores produced by the fungi, algae, and protozoans. Fungi like Plasmodiopora, Mycota, Mycota, Chytidia, Mycota, Myxomycota generally produce these zoospores. Aplanospores means very simple. They are spores with no flagella. So if it is a flagella is there, we can call it as zoospore. If there is no flagella, we can call it as aplanospores. Occurrence generally Oomycota and Phycomycetes, we can see this zoospores, whereas in green algae and Zygomycetes, we can observe this aplanospores. And in flagella is generally present in different types of flagella can be observed, but in aplanospores, the flagella is absent. So motility, zoospores are motile because they are having a flagella, right? Non-motile because they are having no flagella is aplanospores. Cell wall do not possess in these zoospores do not possess any true cell wall. They're actually wall-less. In zoospore morphology also I explained that it contains a cell, some kind of a membrane, right? So in aplanospores possess a true cell wall. Size is very small comparatively aplanospores, which are big in size. Food reserves are generally absent. In aplanospores are contains food reserves. Survivability, incapable of enduring the harsh environmental conditions. Once the zoospores are formed, they may cause infection or survive in water for some time. So they cannot survive for long time or unfavorable environmental conditions. But aplanospores are capable of enduring the harsh environmental conditions. Generally, they are produced by, as we discussed, Oomycota, Chytidiomycota, Myxomycota group of fungi. So generally, the aplanospores are produced by again lower fungi also, but you can also very simple. If the spore having flagella, it is zoospore. If the spore having no flagella, it is aplanospore. These are all the general, understanding for zoospores where it contains flagella for locomotion. So when it comes to aplanospores, we can see very strong outer coat and a very strong nucleus. These are the aplanospores of Spirogyra. So this aplanospores helps to survive unfavorable time. And difference between sexual reproductions, that is a plasmogamy and a karyogamy. So in plasmogamy, two protoplasts are actually bringing their nuclei in a single cell. That is called generally plasmogamy. In meiosis is generally a reduction division where fusion of meiosis, not meiosis, karyogamy, right? So in karyogamy, two haploid nucleus are fused to form a zygote. Generally, we can observe in Zygomycota group of fungi where the two haploid nuclei come in contact and they fuse to form zygote. And other names are plasmogamy is dikaryoticization, whereas the karyogamy is known as the diploidization. After fusion, the two haploid cells N plus N, two haploid cells fuse to become a diploid 2N condition, right? So here the location of process plasmogamy generally takes place in a cell. When it comes to karyogamy, it happens in a specialized organ called zygotes. Nuclear fusion in plasmogamy, nuclei are not merged during plasmogamy. When it comes to karyogamy, nuclei both are merged. If you want to form zygote, both need to merge, right? So that is what happens in karyogamy, but in plasmogamy there is no merging. Result in cell. So this plasmogamy is again N plus N condition only, but it is in diploid. As I told you, two haploid will fuse to form a diploid, and cell containing two haploid nuclei again N plus N, that is called a dikaryotic. The cell containing diploid nucleus, one diploid nucleus. So zygote formation, no zygote formation can be observed in plasmogamy. When it comes to karyogamy, zygote formation takes place. So the followed by this plasmogamy is immediately followed by karyogamy. This karyogamy is immediately followed by meiosis. So in sexual reproduction, three events are there: plasmogamy, karyogamy, and meiosis. So in site of syngamy, so the plasmogamy is the first stage of syngamy. So plasmogamy is actually the second stage of syngamy here. And again the difference between zoospores and zygospores. Zoospores are the again same naked motile spores that is actually produced inside the sporangia. So these zygospores are produced during the fusion of two similar fusion of two similar gametangia. Here sexuality, these are all the zoospores are the asexual spore, whereas zygospores are the sexual spores. Motility, they are motile because they are having flagella. So zygospores are non-motile because it possesses no flagella. Spore wall, it is an actually naked and contains no significant cell wall, but this zygospore contains significant cell wall since it is the resting spore. And flagella, there are 4 types of flagella can be observed. Like anti-retensile, posterior whiplash, such kind of flagella we have already seen the types of flagella and flagellation. But when it comes to zygospore, no zygospore contains flagella. Nuclear condition, this is generally zoospore is a haploid, always haploid. Zoospores are not only zoospores but also aplanospores or planospore, all are haploid only. This zygospore is generally diploid. When it comes to nuclear division, it undergoes mitosis and produce one haploid generation. When it comes to zygospore undergoes meiosis that will produce next haploid generation. So the color generally zoospore is hyaline in color. When it comes to zygospore, it is a little bit brown to dark brown in color or black color. Resistance, less resistant to unfavorable conditions. As I told you, it can the survival period is very less when compared to other spores. It is actually formed during unfavorable time to survive for long periods. Examples: members of Omycota can produce Zoospores, whereas members of Zygomycota can produce Zygospore. Here there is a specialized structure which are present in either side of the spore is known as suspensor. So middle some echinolate structure will be there, that is called Zygospore. That was handled or anchored by either side by a special organ is called suspensor. And now the difference between sexual reproduction and asexual reproduction. In sexual reproduction, fusion of male and female gametes takes place, but in asexual reproduction, it doesn't involve the fusion of male and female gametes. So gametes are male and female are produced, but there is no such kind of gametes are produced in asexual reproduction. Mating types, 2 mating types are present, male and female, but here it requires only one kind of mating type. Nature of the process is a very slow process, but it creates a great genetic variability. It is a very fast process, but it produces almost no genetic variability. Meiosis and mitosis, only meiosis occurs during the divisions. Here only mitotic divisions occurs. Genetic variation, sexual reproduction can enhance actually the great genetic variation as I discussed. So it will produce very less genetic variations, and the occurrence of chances of mutation is also very less. So sexual reproduction is highly useful for natural selection and evolution, but whereas asexual reproduction is which evolves, so no species. Here variation is produced. In asexual reproduction, no variation is produced. So this sexual reproduction is achieved by fertilization, whereas this asexual reproduction is achieved through budding, fragmentation, and sporulation. And to in a exam point of view, there is another names of this phylum that we have discussed. Generally oomycete is otherwise called as egg fungi or pseudo fungi or zoosporic fungi because they will produce eggs, right? It is not a true fungi, that's why it is known as pseudo fungi. They produce zoospores, that's why it is called as zoosporic fungi. Zygomycota are otherwise called as yolk fungi or zygote fungi because the zygote is formed. Ascomycota generally called as crown fungi because it is one of the biggest fungi or biggest classification or many fungi that comes under this. And sac fungi because it will produce a sac-like structure. Basidiomycota is called as a cup fungi because where the basidium is formed above the cup shaped. Club fungi because it looks like a club. Deuteromycota is dustbin fungi because fungi imperfecti are a fungi, dustbin fungi are fungi imperfecti because it contains no sexual stages. Downy mildew fungi is known as hairy fungi. Powdery mildew fungi is known as sac fungi. Actinomycetes fungi is known as ray fungi. When it comes to Pezizaceae, generally it is called cup fungi. So this slide is all about exam preparations. Other names of fungi, sometimes they may ask exams. So this will be, hope it will be useful. And one more thing, a student used to confuse where asexual stages are otherwise known as anamorphic stage, otherwise called as mitotic stage because it mitotic reproduction will happen actually, and otherwise called as imperfect stage. All are gives the same meaning only. Residual fungi are secondary fungi. I repeat, asexual stage otherwise called as anamorphic stage, mitotic stage, imperfect stage. When it comes to sexual reproduction, it is called teleomorphic stage, otherwise called as meiotic stage, because meiosis will happen in sexual reproduction, right? Otherwise called as a perfect stage. So here the sexual spores are in the sexual reproduction, sexual spores are produced through the process of meiosis. That's why it's called also called as meiospores. We can take the example of Zoospore, zygospore, ascospores, and basidospores are the examples of sexual spore. Zoospore and zygospores are the diploid resting sexual spores, whereas ascospores and basidospores are the haploid non-resting spores. Asexual spores are otherwise called as mytospores because they developed through the process of mitosis. So examples are Zoospores, Conidia, Oidea, Spermatia, Microconidia and Macroconidia. These are all the examples of asexual spores. This is also have a great importance related to exams. They may ask in various dimensions. So having a general knowledge about all these terminologies or other names will be greatly useful. For more information, students can refer my book, A Vision into Plant Pathology, a complete student version, which is available in Kalyani Publishers. For further doubts, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.